All right, hi everyone. I hope you guys are doing well, staying warm. So this is a lecture video of the material that we would have covered in class on February 16th. So I'm going to send you guys an email about this, but basically I made the call that it would not be safe to expect you guys to travel into class tomorrow, so we needed to do something virtual. But then when considering my options there, I know it's possible that the power may go out. So in the event of a power outage, WebEx could be interrupted. So I decided to go ahead and record a video ahead of time and post that for you guys on YouTube. So we will have an activity that I'm going to be setting up for you on Canvas the same way that I did Activity 5. So remember that last Thursday, Activity 5 opened in Canvas, and it's going to be open until the end of the day on this coming Thursday, which will be February 18th. When I say the end of the day, I have it set to close at 11:59 p.m. So I'm going to do the same kind of thing with the activity for today's lecture, which is going to be Activity 6. I'll set it up to open on February 16th and then to be open for a week. All right, if you have any questions about anything, let me know. The other announcement before we get started is that Exam 2 is going to open right after class on Thursday. So theoretically, I guess we could be back in the classroom on Thursday. At this point, I'm recording this on Monday afternoon. I don't know for sure if we'll be back in the classroom on Thursday, so look for email announcements about that. All right, so I appreciate you guys being flexible with me. Let me know if you have questions. So the last time I talked with you guys, we started the slides on Chapter 7. We were talking about the development of the nervous system. So we went through the different stages of the nervous system, and then we'd started talking a little bit towards the end of class on Thursday towards the end of lecture on Thursday, about some things that can cause intellectual disability, and we were talking about fetal alcohol syndrome, etc. So picking up where we left off, when we're talking about the development of an individual, we have to compare and contrast genotype versus phenotype. So your genotype is all of the genetic information that you have from biological mom and biological dad. So you have 23 pairs of chromosomes, which means you have 46 chromosomes altogether, half from biological mom, half from biological dad, and the sum of all of that genetic information is your genotype. On the other hand, the phenotype would be the physical characteristics that we can see. So what do you look like? What abilities do you have? What sort of personality do you have? Now, your phenotype is influenced by your genotype. Certainly, your genetics do impact your unique characteristics, but also your phenotype is impacted by the environment as well. So we're going to talk a little bit about this more coming up, but if you think about identical twins, they may have the exact same genotype if they're identical, but they probably would not have the exact same phenotype. There would be some differences in personality, preferences, abilities, and that's because even if they have identical genetics, they probably had at least some aspects of their environment that were different. So genotype is strictly biological. Phenotype includes both biological and environmental characteristics. Okay, now, when we were talking about different causes for intellectual disability last time, we said that intellectual disability means that an individual has a low IQ as measured on an IQ test, and that a person has some sort of dysfunction. So maybe issues with self-care, with academics, with relationships, etc. So we talked about fetal alcohol syndrome, and that is the most common preventable cause of intellectual disability. But if we're thinking about some other potential causes, Down syndrome would be one. So with Down syndrome, you have this extra chromosome. Sometimes it's called trisomy 21. So as I said, you have 23 pairs of chromosomes, so 46 chromosomes total. What happens with Down syndrome is that you have an extra copy of this 21st chromosome, so you have 3 there where you should have 2. This can cause a variety of symptoms. There are certainly unique factors in each case. Not every person who has Down syndrome is the same as every other person who does, but you tend to see intellectual disability, and there can be other psychological conditions, anxiety, that sort of thing as well. But then also some physical characteristics. So there's certain, facial features, usually shorter. So there's an impact on growth. There can be heart-related conditions, etc. Another disorder here, Fragile X syndrome. So it talks about inheriting extra trinucleotide repeats. Okay, basically what we're saying is that there is a portion of the X chromosome that can be fragile, and that if you have these repetitions, if you have too many of them, then it can cause Fragile X syndrome. There is some variety as far as how many repeats the typical person has. You don't have to know this, but I think your textbook says somewhere between like 6 to 50 repeats is okay. But if you get somewhere closer to the neighborhood of 200 repeats, that's where you start to have a problem. So that causes this portion of this X chromosome to be damaged. Things that can happen as a result here, there can be some differences in appearance, so facial features, etc., intellectual disability, other psychological conditions, behavioral issues, ADHD, etc. Now, one thing to think about here, if we have damage to this X chromosome, that's going to impact biological males and females differently. So we're going to talk a little bit about sex determination in the next set of slides. But we know that XY chromosomes are associated with being a biological male, XX chromosomes associated with biological female. So if an individual has XY chromosomes, then there's only one X there. If there's damage to that X, then that's probably going to cause a greater issue because there's no healthy X to provide support there. So Fragile X syndrome is something that we see more in biological males. It's more likely to happen in males, and then the symptoms tend to be more severe in males as well. Okay, another disorder here that's a really interesting interaction between genotype and phenotype. So Phenylketonuria, long name, for a disorder that happens when you don't have the enzyme that's needed to break down the amino acid phenylalanine. So what happens is that the individual will take in a lot of phenylalanine just from a typical diet, but because their body is not able to break it down, then it can build up to dangerous levels and cause intellectual disability. That's one of the prominent symptoms here. What's interesting is that your genotype may cause you to have PKU, but by altering the phenotype, by altering the environment, we can change whether there's any actual distress or dysfunction here. If the person has a special diet, then there's no reason that they would go on to have intellectual disability. Now, then the question is, how do we know that a person has PKU? Because, and I'm not an expert here, but there's a certain type of formula that a baby needs in order to avoid damage here if a baby has PKU. So this is something that is part of standard care. Giving birth at a hospital, then baby would have a blood test. It's extremely rare. Somewhere like 1 in 10,000 babies are born with PKU, something like that. But it is something that we can easily test for. So it's part of standard care to make sure that a child does not have PKU, because now that we know about it and we can screen for it, then you can prevent any dysfunction as a result of that. All right, now, when we're talking about nature versus nurture and the development of the nervous system, this can lead to a conversation about clones. So when it talks here about clones being genetically identical animals, sometimes this happens naturally. If you have identical twins, then technically you have clones. They are genetically identical. And we'll talk a little bit more about twins coming up just a bit. And I don't remember if I've asked if I have any twins this semester. This is where, if we were in the classroom, I would ask you guys to share, if you wanted to share, if you're part of multiples. But what about clones that are not naturally happening? So some animals have been successfully cloned. So I haven't looked up the extensive list. I know sheep and other animals have been cloned. The way that works is that you would take a cell from the individual that you want to clone, and you would take a somatic cell, which means any cell in the body that's not egg or sperm, because your egg and your sperm is only going to have 23 chromosomes, but you want to get a cell that has all 46 chromosomes. And then you can extract the DNA from the nucleus of that cell and then implant it into an egg. So you would have also taken an egg and removed the DNA from that egg and then put in the DNA that you got from your sample, and then you could implant that into a surrogate, And then theoretically, you could have a clone where the new individual would have the same genetic information as the individual who you got that DNA from. Now, certainly, if we were in class, perhaps we could debate the ethics of this, but that is something that has been done in animals, all right? Another thing to think about here when we're talking about genetics, you see epigenetics, the study of factors that affect gene expression. So with epigenetics, we're not changing the genetics that you have, but we are making certain genes more or less likely to be expressed or to be activated. Not actually changing the genes themselves, but certainly you have several different components to your genotype, to your genome, and some of them may be more or less active. And so there are some things that we can do to activate certain portions of genetic material. And one thing that's associated with this is parenting. Now, your textbook focuses on mothering. Oftentimes, the research on parenting does tend to focus on mothering. Certainly, I think that fathering is very important as well. But focusing here on mothering, what they have found is that the type of mothering that an individual receives plays a role in which parts of their genome are expressed. Not changing the content of your genes, but changing whether certain genes are more or less likely to be expressed. You see the term methylation here. Methylation makes a particular gene less likely to be expressed. So here's the way this works. They've done research on this in animals, but then they've also replicated this somewhat with humans as well. If an individual receives poor mothering, so the mother is abusive or neglectful, not providing for the offspring, then that individual undergoes this methylation process, which makes the stress response gene less likely to be activated. Basically, what happens is that the part of your genes that are built in to keep you from being overwhelmed by stress are not as active, which causes a heightened response to stress. On the other hand, if the individual received ideal mothering, then this methylation would not take place. And then the part of your genes that helps you process stress would be healthy and functional, and the individual would not be as heightened in their stress response. Now, certainly, there are tons of different things that can impact the way you handle stress, and mothering is not the only one, but it is one that research does support. Okay, so what we're saying is that you have genetic information, but the environment can impact which part of your genes are actually activated. So this is where we get back to the idea of twins or multiples. So I am not a multiple, and I don't really know anyone well who is a twin or a triplet or something like that. But I have had some students in the past talk about this a little bit. So when we look at identical twins, they come from one egg, one sperm that's split into two, or three if you're talking about triplets or more. So they have identical genetic information. So their genotype is the same. But they often have at least some subtle differences and sometimes not so subtle differences in personalities, preferences, abilities, etc. So then the question is, why is that? Why is it that identical twins are not always identical in their behaviors? And part of this has to do with the difference in environment. Now, if you are a twin, then you probably have very similar environments with your other twin, probably. Certainly, there's been research on twins that have been separated at birth and that kind of thing, but that's relatively rare. Usually, you do have some similarities, but you may have some differences in the way that your parents interact with you and the way that peers interact with you that could potentially cause, personality differences. So while we do tend to see a lot of overlap in identical twins with behavior and outcomes and personality, there certainly are some things that are unique, and that's probably because of the influence of the environment. Another way that we can see the environment impacting your development has to do with a sensitive period. So a sensitive period is a time during development when you can influence something and cause a permanent alteration. So you have, for example, a sensitive period for language. And during that sensitive period, being taught language, having someone speak to you, read to you, explicitly teach you, can cause you to learn language that will stick with you going forward. On the other hand, if you're outside of that sensitive period, then it's not likely to stick with you. I mean, if you're trying to teach language to someone who is before their sensitive period, I mean, I don't really know if talking to the individual in the womb is super helpful, or if you're trying to work with someone outside or after their sensitive period. Sometimes there are cases where a child has undergone, terrible abuse, neglect situation where they're not exposed to language. And then there's a point where even if a person is trying to work with them, they're not able to acquire language. They're outside of that sensitive period. It's already passed. So there's been some research that somewhere around age 7, if you have not acquired a language by that point, you may not be able to. So this is a time period during development when there's an influence from the environment that can cause a permanent change. Another way they do this research has to do with vision. So it talks about monocular deprivation. Once again, we had our conversation about animal research. Certainly there are ethical concerns here. But the way they do this research is with dogs. And what they would do is that they would, at various times during the dog's life, they had, different groups that were different ages. They would deprive the individual of visual experiences in one eye. So you can, like, for example, they could sew the dog's eye shut. I know it sounds awful. It makes me flinch a little bit talking about it. So the idea is that if the dog has already passed their sensitive period for vision, then their vision is already established. And so while the dog is not likely to be happy about having one eye sewn shut, then when you take the sutures out, you probably won't have a significant difference in the dog's vision. On the other hand, if you sew an eye shut during the sensitive period for the development of vision, then that eye is not having those visual experiences in the environment that are so important for proper development of vision. And so after you remove the stitches, then there will probably be some detriment to vision there. So this is an example of a time where if the individual does not have those learning experiences during a sensitive period, then they may not be able to develop that ability or that trait. Now, as we're thinking about development as a lifelong process, right? Development is something that begins at conception and ends at death. It's an ongoing process. So if we're thinking about as we get older, what tends to happen? Well, one thing that we do typically see is that there is a loss in memory as the person gets older. This can be disconcerting. A person may be worried about whether this means that they have dementia, for example, which we'll mention in just a second. But right now, if we just think about normal aging, we do tend to see that the hippocampus shrinks as individuals get older. So you have some cells in the hippocampus that are dying, and as a result of that, you can have losses in delayed memory. This would be considered typical. As a person gets older, the hippocampus decreases in size, and delayed memory can start to become an issue. When I say delayed memory, maybe I give a few words to a person, and then I have them do some other tasks, and then I say, can you remember those 3 words that I told you earlier? To see if they still remember them. On the other hand, immediate memory does not decline in a healthy aging individual. So I'm going to tell you 3 words, and then very shortly afterwards ask you to repeat those 3 words. We would expect that a healthy older individual would still be able to do this, right? So as a normal part of aging, we can see a decrease in memory and a decrease in the size of the hippocampus. But the memory issues tend to be more with delayed memory rather than with immediate memory. We can also see some changes in motor skills. And what's interesting is that some of your cells that control motor abilities do tend to reduce as you get older. It talks about the Betz cells in the motor cortex that start reducing after age 50 and then may be completely gone in the 80s. On the other hand, it talks about inferior olive cells here now in the brainstem, so a little bit deeper in the hindbrain area, that are also important for motor function that do not decrease as a person has normal, healthy aging. So this is where we can see a loss of some, but not necessarily all, motor control. Of course, there's a conversation to be had here which maybe if I had you guys in front of me right now, we would have a little bit about driving in older individuals. At what point should you not drive anymore? Certainly there are some concerns, decreased reaction time, more likely to be confused and that kind of thing. Motor skills maybe decreasing, but not completely. I don't think there's a good answer here about when an individual should stop driving. Certainly there's not a universal answer as if this is the age where you need to do that. I do have older parents, and the last time that my dad got his driver's license renewed, he told me, I got it renewed for 8 years, so you can't take it away from me for at least 8 years. He knows that's not how it works, but it's certainly not a conversation that I'm looking forward to engaging in. All right, so it is normal as a person gets older to have some decline in motor skills, to have some decline in delayed memory, to have a reduction in the size of the hippocampus. But we can have some issues that come up that are kind of over and beyond what we would expect based on typical aging. So when you see the term dementia, dementia is kind of a general umbrella term here. So we've had a major loss of cognitive function. Now, dementia can be caused by a variety of different things. When you see the term senile dementia, that means that it's dementia caused by old age, right? But there are other types of dementia as well. Sometimes cardiovascular issues can cause dementia. If you're not getting enough oxygen to your brain, then that can cause a person to lose cognitive abilities. And if you're able to treat that relatively quickly, then those abilities may come back. On the other hand, with senile dementia, as a person gets older, we don't really have great treatment options for helping the individual get back here. So Alzheimer's disease is one disease that would fall in that category of senile dementia. I've mentioned in class before that one of my grandfathers passed away from Alzheimer's disease. So I have seen firsthand how cruel this disease can be. So Alzheimer's disease usually starts off with memory loss that might be confused with typical aging. It is normal to have some loss of that delayed memory, but then the memory might be a little bit more immediate, a little bit more severe, the decline a little bit sharper than we would typically expect. It talks about the brain showing reduced metabolism. So remember that your brain is very needy. It needs a lot of oxygen, it needs a lot of nutrients. And in individuals who have Alzheimer's disease, their brain is not properly managing those resources. And then cortical atrophy, so you can have cells in different areas of the cerebral cortex that can start to die, and that is associated with memory issues. Now, when we talk about Alzheimer's disease, it's important to note that we don't have a firm answer on the cause yet. There are some genetic components, but certainly if you've had a family member who has had Alzheimer's disease, it does not necessarily mean that you will. There are environmental things that you can do to reduce risk as well, things like exercising and staying cognitively active and taking a vitamin. There's been some discussion of like omega-3 supplementation, that kind of thing, which couldn't hurt really, but talk to your doctor about that. So with Alzheimer's, what we found is that you tend to find these senile plaques and the neurofibrillary tangles. And so it was thought for a while that these were the cause of Alzheimer's disease. So with the senile plaques, we're going to talk a little bit more about this coming up. But you have these beta amyloid, this little symbol here is beta amyloid buildups here that cause plaques that are found between neurons that disrupt the connection between neurons. And then neurofibrillary tangles, so you have these neurofilaments that sort of help form the structure inside of the cell. And when these neurofilaments become tangled, then that can damage the cell and the cell may die. So it talks about basal forebrain nuclei disappearing. So remember that we're in the central nervous system. So a group of neurons here would be a nucleus. If there's just one, nuclei is plural. So when you see nuclei, that's a group of neurons in the central nervous system. The basal forebrain is towards the bottom of the forebrain. You can see a picture here, the basal forebrain. So one thing that's interesting with Alzheimer's disease is that in order to confirm Alzheimer's disease, we need to know for sure that an individual has these plaques and tangles. You can see in these pictures here, I believe the yellow things are the tangles and the darker things are the plaques. I think that's right. So Alzheimer's disease is not something that can be 100% confirmed unless an individual has passed away, and then on autopsy we find these tangles and plaques. We can diagnose suspected Alzheimer's disease during life, but to confirm we need an autopsy there. Okay, so here's the thing. We have these beta amyloid plaques that are building up, and it talks a little bit about this process here, right? So we have this amyloid precursor protein that's being broken down, and one of the byproducts of that is beta amyloid. So it's normal to have beta amyloid in your nervous system, but the beta amyloid is supposed to be cleaned up here. One important, it talks about apolipoprotein here, is an important factor in breaking down the beta amyloid. So it could be that a person has too much of the amyloid precursor protein. So there's too much of this beta amyloid. Or it could be that a person does not have enough of this apolipoprotein to break down the beta amyloid. So there have been treatments that have been targeted at trying to decrease beta amyloid or to increase the proteins that break down beta amyloid. Unfortunately, those treatment options have not produced the results that we had hoped for, which then leads us to wonder, when we see the plaques and tangles, is that the cause of the problems, or are plaques and tangles just a symptom of another cause? Because when we treat the plaques and tangles, it doesn't reduce the symptoms as much as we would hope. A lot of the work with treating Alzheimer's disease has focused on acetylcholine, right? That's a neurotransmitter that's important for learning and memory. And so by decreasing the enzymes that break down acetylcholine, then we can have more activity from the acetylcholine. But that's something that is only helpful for a limited period of time. It's not a long-term fix. So certainly there's hope that we will come up with better treatment options for that. Okay, switching gears a little bit here. Chapter 12 talks about the biological basis of sexual activity. So this chapter is really split into two sections. In the first section, we talk a little bit about reproductive behaviors. And then in the second part of this chapter, we talk about the development of sex characteristics. And then we'll talk a little bit about certain disorders that can happen with there, etc. Now, my plan is to cover maybe half of these slides, or a little less than half perhaps, today, and then to finish this up in class on Thursday. Hopefully we'll be back on campus on Thursday the 18th. Okay, so switching gears here to reproductive behavior. 4 stages of reproductive behavior. First of all, it talks about sexual attraction. You can see we have some examples going on over here with birds, right? So we got peacock, peahen here. So we have sexual attraction, appetitive behavior, copulation, and post-copulatory behavior. So we're going to go through and talk a little bit more about each one of these. So what you'll see is that you have these plus arrows and these minuses. So plus is anything that makes sexual activity more likely to happen. So sexual attraction is going to lead to an appetitive behavior, which we'll define coming up, is something that increases the likelihood of sex. But then after having sexual activity, then the post-copulatory behavior sort of has a decrease in sexual activity. It's less likely afterwards. There's a refractory period here that we're going to be talking about before this cycle starts over again. Now, one thing to mention here is that your textbook is looking specifically at reproductive behavior here. So here it's talking about sexual activity between like heterosexual individuals, because it's looking at the reproductive behaviors. Certainly that's not the only situation in which sexual behavior occurs, but that's the one that your textbook zeroes in on at this point. So when we're talking about the different stages here, we start off with sexual attraction. This can be synchronized by a few different things. One thing, it talks about physiological readiness to reproduce. So for some species, this is especially important, that maybe there'll be some signals that are being released, some pheromones, certain behaviors that indicate that an individual is ready to reproduce. And perhaps that is part of what is found sexually attractive is that there's some kind of signal that that individual is ready to reproduce. But it also talks about learned associations here. So this is where we could spend some time, if we wanted to, thinking about how we've been classically conditioned to find certain traits attractive or to find certain behaviors attractive. So maybe this is what we learn from society. This is what we learn from experiences as far as what we find attractive there. This, because it's coming more from the environment, can change a little bit more over time. For example, in my interpersonal relations class, we talk about how there's a connection between food supply and the type of body style that's considered most attractive, that heavier individuals might be considered more attractive in cultures where the food is scarce because it's showing a sign of like health and access to resources, etc. So some of this can change over time from situation to situation. I think it's interesting, your textbook talks about how some species have pair bonds that live together for long periods of time, maybe even for life, which if we were in class, I would ask you guys if you think humans fall in this category. Certainly, hearkening back to my interpersonal relations class, our divorce rate is about 50%. So certainly this can happen in humans, but it doesn't always. But, there are some animals that mate for life. There are a lot of birds. I think about birds, but I'm sure there are other examples of animals that mate for life as well. All right, this is where we get to activity 6. So there's a note here, and I think this part with the definition for repetitive behaviors is probably in your notes. And then the activity is probably something extra that I've added here for these slides. So I'm going to set up activity 6 the same way that I set up activity 5, where it'll open on February 16th. You can go in, submit it that way. It'll be open for a week. So the activity here is asking you, what are some examples of appetitive behaviors in humans? So we start off with sexual attraction, and then we move into appetitive behaviors. Appetitive behaviors are really any behaviors that are intended to increase the likelihood of a sexual interaction. So this is where, if you're thinking about examples in humans, what do humans do? Is there a measure of gift giving or hygiene or clothing? Like thinking about what humans do to increase the likelihood of sexual interactions. And certainly I look forward to reading your responses. All right, so let me know if you have questions about that. So somewhere along the way, my life took a turn where now I am teaching college students about coitus. I don't know where that happened, but it's cool. I like my job. All right, so copulation, coitus here. It's a term that involves one or more intromissions in which the male penis is inserted into the female vagina. So there you go, right? I'm sure you got this. I don't need to have any further conversation about this, right? Okay. Once again, I just want to clarify that your textbook is zeroed in on reproductive behavior here, which is why it's emphasizing this, male and female component. One thing to think about here, it talks about the male ejaculating semen. So is it necessary for a man to have an orgasm in order for pregnancy to occur? Yeah, pretty much. I mean, I suppose there's a possibility of some fluids being released beforehand, but pretty much that's the case. Does a woman have to have an orgasm to become pregnant? Not necessarily. There's some research that suggests it might be helpful. If you think about the muscle contractions that can come along with that might help usher the sperm into the correct location, but certainly not absolutely necessary. Okay, so after copulation, a refractory phase follows. Perhaps when we were talking about action potentials, you guys were thinking about this, but you were nice enough not to mention it at that point. When I talked about with a refractory phase, so you have your action potential that occurs, and then you have this absolute refractory phase and relative refractory phase where your neuron is not able to fire again, or it's less likely to fire again. Not a very different concept here with refractory phase after copulation. You'll see this again on a future slide, but when we're talking about refractory phases here, we're talking about males and an inability or a decreased ability to engage in sexual activity again following orgasm. Okay, Now, there's a term here it talks about in your textbook. If a female is willing to copulate, she is sexually receptive in heat, estrus. Okay, your textbook does a lot on animal research. You've sort of gotten that theme, perhaps. And your textbook has a lot about animals in this chapter as well. I took a lot of it out because I didn't think, having conversation about rodent reproduction was just super helpful. There'll be a little bit sprinkled throughout here, but I think we usually use these terms, like in heat and estrus, when we're thinking about animals, and can certainly seem derogatory, demeaning to apply these to humans, for sure. Okay, but if we have copulation, which is the actual sexual activity, and then we have post-copulatory behavior, what happens after the sexual activity is finished? There are many different things that could fall in that category, but technically, parental behaviors would be considered post-copulatory behaviors because sexual activity can lead to reproduction, that can lead to the need to parent. This is from your textbook. I just had to leave this in here because I think it's just so hilarious. Guinea pig Don Juan. Okay, so Sooty, male guinea pig, escaped from captivity and then re-entered captivity in a nearby cage housing 24 females. 2 months later, he is now the father of 43 offspring. We did a head count and found 25 guinea pigs. Sooty was fast asleep in the corner. He was absolutely shattered. We put him back in his cage and he slept for 2 days. So that was his post-copulatory behavior. But certainly this could vary from individual to individual and species to species. So I hope you guys enjoy the guinea pig reference. This is really a lecture where I miss having you guys here to at least see your faces and how you respond to some of this. Okay, so when we're talking about parenting behaviors and talking about reproduction, we should clarify different types of fertilization. So with fertilization, we're talking about gametes coming together, sex cells, so sperm and eggs. When they come together, we have a zygote, right? Fertilized egg. Now, you can have internal fertilization or external fertilization. With internal fertilization, this is what we see in mammals and in some other species as well, but mainly we think about mammals here, where the fertilization takes place inside the female's body, versus external fertilization, where maybe like we lay eggs and then the sperm fertilize the eggs. Now, there are a few different pros and cons to the different types of reproduction here. On the one hand, with internal fertilization, if we are, if mom is carrying the babies, then perhaps that means that there's more protection, there's a more stable environment, stable sources of nutrition, care after birth, that kind of thing as well. On the other hand, with external fertilization, there are advantages here as well. With external fertilization, you can have a lot more. Like, there's a limit to internal fertilization. I don't personally know the most babies that a woman has delivered. Like, I know, like, there was Octomom, right? There was a mom that had 8 babies at once. I think that was right, but I don't know. I haven't looked into this, but there's certainly a limit. Typically, we think about humans delivering 1 or 2 babies at the most, and then occasionally more. But with external fertilization, you can have a lot more eggs. But then, of course, there's not that protection there. If you think about Finding Nemo, then certainly there can be situations where the protection would have been helpful. So I suppose internal fertilization is more about quality and external fertilization is more about quantity. Okay, now, when we're talking about reproductive behaviors, we know that hormones are important. And we'll talk a little bit about hormones in females, but a lot of the research on the importance of hormones in reproduction is focused on males. So when a male is castrated, that means that in some way he has lost the use of his testicles, which could be like a physical, like removal, could be like a chemical situation, but where a male's testicles are no longer functioning, which means that testosterone is not being produced. So when an individual has no testosterone, then he, if we're talking about a male individual here, would not have interest in sexual activity because the testosterone is not there. What we found, though, is that there can be an activational effect. when you see this activational effect, it means that it's going to be brief. So if you take an example, your textbook talks a lot about mice, so why not think about that? If you take a castrated mouse that has had either physically removed or chemically, the testicles are not functioning here. So testosterone is not being produced, so this mouse is not interested in mating behaviors. And then you inject the mouse with testosterone. You will have a brief re-interest. The interest will be rekindled in the mouse for a limited time until that hormone shot wears off, so until the testosterone is excreted from his system. And then he would go back to that testosterone level of sexual interest. So this is an activational effect because it only lasts for a short period of time. Now, one thing that we see is that hormones certainly are more complicated in women because women go through cycles that men don't go through. So when we're talking about cycles in females here, certainly women do have testosterone, but they have lower levels of testosterone on average than men do. So women going through cycles here. So if you think about a female menstrual cycle, the stereotype standard number that's often used is 28 days, although there's a great level of variation there. But basically saying like day one of the cycle is when a woman starts her menstrual cycle, starts her menstrual period, and then that lasts for however many days that lasts. So about halfway through the cycle is usually when ovulation takes place. And so the hormones are going to be preparing for this process, having an egg that is prepared and then released. And so during ovulation, that egg is in the ideal location for fertilization to take place. Now, then the question is, can you predict ovulation? Yes. There are tests that you can take that are similar to pregnancy tests that measure the hormones that are associated with ovulation. So yes, you can take a test that will tell you whether or not you're ovulating. Should you use that as a reliable birth control method? Not necessarily, because sperm can live inside the body for several days. So if a person ovulates later, then they could still get pregnant if there were sperm that were still in their system. So certainly this is not a guaranteed method. But if someone is trying to get pregnant, then you can use the ovulation predictors to help with that. Okay, now in addition to hormones like your testosterone, estrogen, etc., pheromones are chemical signals that are used to communicate information. So when we think about pheromones, we often think about animals that release pheromones to let other individuals of their species know that they are interested and available for mating. The question is, do humans use pheromones? And very likely the answer to that is yes. One research study, I realize this is going to sound kind of odd, but one research study, they had women wear some sort of pad sort of thing under their arms, and then they tracked like where a woman was in her menstrual cycle, and then they had male individuals smell the pads. Okay, so the women are not there, they're just smelling the essence of the individual. And they found that the men reported the women who were ovulating tended to have a more pleasant smell to the men. They chose, like, which one of these do you think smells the best? They would choose the woman who was ovulating. So, yeah, kind of an odd thought here, but perhaps there is something going on with pheromones here as well. Also, it talks about pheromones conveying other information as well. Your textbook talks about with elephants, like elephants producing certain pheromones to show, like, for example, which one is the alpha, which one is the leader, versus which one is more like submissive. And so this can help reduce fights and that kind of thing. Now, when we're comparing human sexual behavior versus behavior in animals, we tend to see that there are a lot of similarities, but humans may engage in a larger variety of sexual behaviors, and also humans can tell us a lot more about it. So this is where the subjective reactions such as orgasm, we can get more of a description from humans than we can from other animals. I have never taught human sexual behavior. You know that the grad students are teaching that right now. That's totally fine. That they can have that. I've never asked to teach this class. Certainly, it's an interesting topic, but not necessarily one that I'd like to lecture on full time. When we're talking about human sexual behavior, where do we get this information from? Well, certainly some of this is from surveys. You know, that's where we get a lot of information, but then it's hard to know if a person's telling the truth. There have been studies that have done observational research on sexual behavior. Masters and Johnson, you can look that up if you're interested. One thing to consider, if a person has agreed to be part of this observational study on sexual behavior, will those findings generalize to the population, like to other people who did not volunteer to be part of an observational research study? Yeah, I don't know. But a lot of this has come from the research that Masters and Johnson did. So when we're talking about stages of sexual response, you're going to see a couple of graphs on the next couple of slides, but excitement, plateau, orgasm, and resolution. So we'll take a look at that. One thing that you'll see when we look at human responses, that women tend to have a greater variety of responses. For men, there tends to be a pretty stable pattern here. The exact amount of time that you spend in one section versus the next section may vary, but going through these stages pretty consistently for men. Also, as I mentioned earlier, men tend to have an absolute refractory phase, so that after an orgasm, there is a period of time where it's not possible for a man to engage in sexual activity again. Okay, so here we have a pretty simplistic generalization, but this is what we tend to see with males. So you have an increase in sexual excitement here, and then at some point there's a plateau, so we stay at about the same level, and then there is an increase in sexual excitement that leads to orgasm, and then afterwards you have this refractory phase. During the refractory phase, it would not be possible for the individual to, and it would not be physically possible for an individual to engage in sexual intercourse again until after that refractory phase is ended. After the refractory phase is ended, then it's possible that the cycle could start over again. On the other hand, we can see very different patterns in women. So here are three of the more common patterns, but certainly there could be many others as well. So here's what we're seeing with line A. You know, we have increase in sexual excitement. We have a relatively long plateau here, and then you'll see an individual having one orgasm or possibly two. So this is another difference here, that women are capable of having multiple orgasms versus men. You can also see some women that have a very significant increase in sexual excitement without really having any discernible plateau, and then hitting orgasm very early compared with some of these other options. And then with this option here, maybe an individual has an increase in sexual excitement that leads to a long plateau that does not reach a level of orgasm. And certainly this could vary from person to person, as well as from encounter to encounter, right? Now, why is it that women have more of a variety here in their responses? Part of this is more biological, as in like the absence of the refractory phase, that kind of thing. But also, women report having more of an emotional and cognitive component to their sexual activity. So this doesn't necessarily mean that this is exclusive to women or that all women feel this way, but on a survey, women tend to report that it's not just a physical act as much as it is an emotional act, and that if there's a satisfaction, not just physically, but emotionally as well, then that makes the individual more likely to want to engage in that sexual activity again. Certainly, as I said, this is what people say on a survey, so it could be that this is not completely accurate, but that's the way we get this information. Okay, so let me know if you guys have any questions also. Feel free to email me as we go through this. So we mentioned the importance of hormones, about the fact that a castrated male would not show interest in sexual activity because there's a lack of testosterone. You see the term androgens here. Androgens refers to male sex hormones, and testosterone would be an example of an androgen. But when we're talking about hormones, they only play a permissive role. What we're essentially saying here is that a male would need a certain level of testosterone to engage in sexual activity. But that does not necessarily mean that the higher the testosterone, the greater the sex drive, okay? As long as there is at least that minimum level, then sexual activity could take place. So if we have an individual who is not producing testosterone and we give them a low dose of testosterone, that is enough to create sexual interest. It doesn't take a high dose. And as the dose of testosterone increases, then that doesn't necessarily mean the sexual activity increases as well. So we're saying that it plays a permissive role. You have to have a certain level of testosterone for the male to be interested in sexual activity, but that does not necessarily mean that having more testosterone increases your sex drive. So sometimes they sell products, and I would encourage you to talk to a doctor. But sometimes they have products they'll sell that they talk about increasing your testosterone and about how this will automatically fix everything in your life and give you a higher sex drive and all of that. It might do that, but assuming that individual has at least a little testosterone naturally being produced, then any other effect is probably just a placebo effect more than anything else. All right, we're going to be switching gears in a second to talk about sexual development, but before we get there, one last thing here. When we're talking about parenting, as a post-copulatory behavior, how much attention does the individual need from parents? So this is where we contrast altricial animals versus precocial animals. Maybe you've heard the word precocious. Someone who's precocious is sort of, they're developing early, they're sort of ahead of where you would expect based on age. So with precocial animals, they're born in a relatively developed state. So if you think about a species where maybe you're in a dangerous situation, if you think about like a zebra being born, on a savanna where there are lions around, certainly a baby needs to be able to run pretty quickly afterwards. On the other hand, there are some altricial individuals that are born in a pretty dependent state. So if you think about human infants, a newborn baby, if you've spent any time around a newborn baby, they can cry, which is their attempt to communicate something. Hard to know exactly what they're communicating, but beyond that, they're certainly not able to meet their needs. So more parental involvement is needed there. Okay, now, for the rest of today's video, we're going to be talking about this process of sexual differentiation. And we're going to go through this process today, and then on Thursday, the plan, assuming nothing else happens to change things, the plan is to talk about the different conditions that can happen that can cause this process to be a little bit different. But first of all, sexual differentiation is the process where an individual develops male or female bodies and behaviors. So we'll talk a little bit more about the body aspect today and a little bit more on Thursday about the behavioral aspect. Now, when does sexual differentiation happen? Well, it's a long process. So this is a process that begins at conception, and we're going to talk a little bit about all the different things that can happen during pregnancy, like in utero, and then it continues into after birth, into adulthood. If you think about like puberty and other things that can happen, this is a process that can continue throughout the lifespan. Now, when we're talking about sexual differentiation, that's something that's happening across a long period of time. Your textbook uses the term sex determination to talk about specifically the chromosomes here. So we will talk about some situations on Thursday where this is not necessarily the case, but for the purposes of explaining this process today, if you have XX chromosomes, then you would be considered biologically female. If you have XY chromosomes, you would be considered biologically male. Now, if we think about that, then how does that play a role in who determines the sex of an infant? So if mom only has X's to give and dad has X and Y, then if mom gives an X and dad gives an X, then the individual would be biologically female. if mom gives an X and dad gives a Y, the individual would be biologically male. And we will talk about some exceptions to this on Thursday. So then it is the sperm that's actually determining the sex of the fetus rather than the egg. So this makes me think to like historical situations. I'm a fan of historical fiction. I'm just going to throw that out there, like British fiction. So the whole Plantagenet family kind of leading up to Henry VIII, I find really, some of it's horrifying, but fascinating, the psychology in there. But one thing, Henry VIII, I don't know what you guys know about Henry VIII, but just sort of a crash course here. He had 6 wives. And so his first wife had several losses, several miscarriages, stillbirths, and then she had 1 surviving daughter. And then he divorced her and he married Anne Boleyn, and she had 1 surviving daughter and several losses. And then, anyway, he had 6 wives, and many of them were divorced or killed in a large part because they were not giving him sons, you understand? He needed a son. And what we find, he was blaming his wives for not producing male heirs when actually that is not something that came from the females. But of course, Henry VIII would have had you beheaded for suggesting that. So anyway, whole another conversation. I think it's fascinating. All right, so in the beginning of this process, so we have chromosomes, right? Chromosomal sex, XX, biologically female, XY, biologically male. In the beginning, the individual has indifferent gonads, which means that there are these organs that could develop into ovaries or into testes, and certainly very early on, you're not going to be able to tell. People use ultrasounds to determine the sex of the fetus, and you'll see some pictures coming up, but certainly there is a point where that's easier to do. Very early on, it's really just a 50-50 sort of guess. So what happens is that if an individual has a Y chromosome, there's a portion of that Y chromosome that is called the Sry gene. So the Sry gene sets into motion this process that will lead to the development of the testes. So those indifferent gonads will become testes if you have this Sry gene. If an individual has XX chromosomes, they don't have a Y chromosome, so there would not be the Sry gene, which means that the indifferent gonad would develop into an ovary instead of a testis. So basically what we're saying is that the human body is sort of by default female, unless acted upon. If acted upon, can become biologically male. Okay, so hormones being secreted by the gonads are going to lead to sexual differentiation. And there's a nice kind of summary that you'll see towards the end of our discussion today that'll kind of add, a nice summary of this. So what's happening is that if you have XY chromosomes, then you have this Sry gene that causes the indifferent gonads to become testes. The testes then produce hormones that lead to further masculinization of a person's biological form. Now, when it talks about embryos here having early tissues, remember that embryo is that term that we use up until around 10 weeks. So very early on, the embryo is going to have all the structures needed to become either male or female. It talks about the Wolffian ducts and the Mullerian ducts. You'll see this coming up. But the Wolffian ducts, if they remain, will form into male internal tubing versus Mullerian ducts that form into female internal tubing. And that's something that happens as a result of the hormonal changes that are happening. So if you have a Y chromosome, you have an Sry gene that leads to the indifferent gonad becoming a testis, and the testes produce testosterone that then causes the duct system to develop in a more masculine way. If you have no Y chromosome, you don't have an Sry gene, then your indifferent gonad becomes an ovary, which would not produce testosterone, which would cause the ducts to develop in a more biologically female way. Okay, so here we are at 6 weeks of gestation. So we're still in the embryonic stage here. You can see that we have an indifferent gonad, which at this point we can't tell if it's going to develop into a testis or an ovary. And you see we have Wolffian ducts and Mullerian ducts. The Wolffian ducts, as this is going to be on maybe an upcoming slide, you'll see the Wolffian ducts will develop in males and the Mullerian ducts will develop in females. Okay. At this point, we have a genital tubercle that could become a penis or could become a clitoris, depending on how this process goes. This genital fold could become the outer portion of a vagina or could become the testicles here, or the scrotum. Scrotum's what I'm looking for there. All right, so in females, the Mullerian ducts develop and the Wolffian ducts fade away. Remember, early on, you have all the components you need to become either male or female. In females, without a Y chromosome, there is no SRY gene, so the testes develop. Sorry, we're talking about females. Wow, let me start back over again there. In females, you have XX chromosomes, no Y chromosome, no SRY gene, so the indifferent gonad becomes ovaries, and then the ovaries will be joined by the Mullerian ducts here. The Mullerian ducts will develop because there's no testosterone and anti-Mullerian hormone that we'll mention in a second that would cause a person to become male. So the Mullerian ducts stick around in females, and the Wolffian ducts would fade away in females. The Mullerian ducts would become the oviducts, so like fallopian tubes, uterus, vagina. On the other hand, in males, if you have XY chromosomes, then you have an SRY gene. The SRY gene causes the indifferent gonad to become a testis, which would then lead to the development of the Wolffian ducts, because there's testosterone that's being produced and anti-Mullerian hormone that's being produced. So the Wolffian ducts then develop. I don't expect you to know all the different functions of the different components here, but you have the male tubing that's involved in adding all the different components of semen and then transporting that. So in males, the Mullerian system fades away and the Wolffian duct develops. Okay, here we are at 8 weeks of gestation. Now, at this point on an ultrasound, you're not going to be able to determine the sex of the individual, but here you can see that the indifferent gonad is becoming, like testis would be, singular, testes would be plural here. And we have the Wolffian system that is developing, and the Mullerian system is starting to fade away here. And here, that genital tubercle is turning into more of a penis. Over here, we have an indifferent gonad that is developing into an ovary. We have the Mullerian duct system that's developing, and the Wolffian duct system is fading away. And we have the genital tubercle becoming more of a clitoris here. Now, as we get to 15 weeks of gestation, this is where you're getting closer to being able to tell on an ultrasound the sex of the baby. But certainly at 15 weeks, it depends on what kind of ultrasound. Is this a 3D ultrasound? You'll be able to get a better picture. With just a regular ultrasound, usually they say at least 20 weeks to get a pretty consistent answer on that. So here, about 15 weeks of gestation for the males, we have the testes that have formed. We have the Wolffian system that's formed. The Mullerian system has faded away now. And we have penis developing, scrotum developing. In the female, we have ovaries. We have the Mullerian system that is developing. The Wolffian system is fading away. And here we have the vagina forming. Okay, so I've used the terms testosterone and anti-Mullerian hormone a bit. These are hormones that are being produced by the testes. So if you have XY chromosomes, then you have an SRY gene. The SRY gene causes the indifferent gonads to develop into testes. The testes produce testosterone and anti-Mullerian hormone. The testosterone is going to cause the Wolffian system to develop. The anti-Mullerian hormone, just as the name implies, is going to lead to the regression of the Mullerian system. So here, the ducts that would develop into the uterus and internal vagina and fallopian tubes, those things would regress. Okay. Now, on the other hand, if you have XX chromosomes, then you do not have a Y chromosome, so you don't have the SRY gene. So the indifferent gonads develop into ovaries, which do not produce testosterone or anti-Mullerian hormone. So there's no masculinization of the duct system there. Now, in a male, Testosterone is important for the Wolffian system developing, but it's also important for the external male genitalia to develop. But what needs to happen here is we need a more potent form of testosterone. So you already have testosterone in your system if you have testes, but then 5-alpha reductase is an enzyme that takes that testosterone and makes it into a more potent form, dihydrotestosterone, DHT here. So you need that more powerful form of testosterone in order to form the male external genitalia, penis, scrotum, etc. We will talk on Thursday about a situation where if an individual does not have this enzyme, then you can see a different result. We'll talk about that on Thursday. Okay, so here's a nice summary. If you have, this is from your textbook, if you have XY chromosomes, then you have the SRY gene. If you have the SRY gene, it means the individuals in different gonads develop into testes that would then produce testosterone and anti-Mullerian hormone. The testosterone causes the Wolffian system to develop, and the anti-Mullerian hormone causes the Mullerian system to fade away. Also, the testosterone is converted into a more potent form, DHT, by this enzyme 5-alpha reductase. That DHT is then important for finishing the external masculinization of the genitals. On the other hand, if an individual has XX chromosomes, then there is no SRY gene. So the indifferent gonads develop into ovaries. Now, ovaries do not produce that testosterone, so that's not going to cause the Wolffian system to develop. They don't produce anti-Mullerian hormone, so that's not going to cause the Mullerian system to fade away. So because there's nothing to get rid of the Mullerian system will develop into the fallopian tubes, uterus, inner vagina, right? And then because you don't have the testosterone to be converted into the more potent form of testosterone, then you have the skin forming the outer vagina and clitoris. Okay, so that's a quick overview of this process as described in your textbook. We will talk about some exceptions to this process on Thursday, and we'll also talk a little bit more about behaviors besides just this sexual differentiation. So if you have questions, please let me know. Remember that I'm going to have Activity 6 opening on February 16th, and I'll be open for a week. Let me know if you guys need anything, and hopefully we'll be back in the classroom on Thursday. So I'll see you guys then.